Clinical laboratories are constantly being asked to do more with less. Sample sizes are reducing, which causes the risk of having insufficient sample quantity or quality for an accurate result. But at the same time, the list of markers that are being requested is increasing, and we're putting these samples through very complex laboratory workflows, increasing the risk of sample swaps, generating an inaccurate result because the wrong sample is being reported. My name is Darryl Irwin and I'm the Vice President of Scientific Affairs at Agena Bioscience. And today we're going to cover the Agena sample integrity portfolio and the solutions that Agena is offering to clinical laboratories, particularly those who are running complex next generation sequencing workflows, to mitigate these potential sources of error. Now, just as some logistics, everyone here is muted and the session is being recorded. If you wish to submit a question, you will find a little bubble with a question mark at the top of your screen. You can click that and type your question into the box. We will have time at the end of this session to answer a number of questions, but if we don't get to all of them, we'll make sure that we get back to you via email. So today we're going to cover the challenges with sample handling and Agena's solution for you to mitigate possible sources of error in your clinical laboratory's results. We're going to talk about how this can be integrated into a next generation sequencing workflow and how several of our customers have already done this. And then lastly, we'll talk about the various different solutions on the MassARRAY for the various different applications that your laboratory is running. So much of the chain of custody errors can happen outside of the clinical laboratory during the collection, the labeling, the fixing of slides, the anatomical pathology. But what we can do is help you ensure that there are no mix-ups and errors that occur inside your laboratory, so that you can be confident that no errors have been because of your laboratory itself. So sources of error include incorrect sample being sent to the laboratory, multiple samples with incorrect labeling, contaminated specimens, and of course this causes wasted reagents, wasted time and money due to failed and repeated runs, and it causes questions around your laboratory's ability to deliver accurate results. Now, this is a real risk. And in the Journal of Urology, there was a study performed across prostate biopsies. And in this particular study with a large number of specimens, they identified that 1 in 40 samples had sample provenance issues. So that is sample transposition or sample contamination. And out of those 1 in 40 samples that had this issue, this resulted in 22.5% of them, or a quarter of them, delivering clinically meaningful false diagnoses to patients. The estimated effect of this was greater than almost $880 million to the health system, or $3,776 per prostate cancer diagnosis. So solutions for a next generation sequencing workflow. Now, if you're running a next generation sequencing workflow, you'll be well aware that there are multiple touch points in this workflow. Frequently, it occurs over multiple days with different technicians dedicated to the different steps. There is a significant investment in your reagents and consumables for the workflow. And there is, of course, an extensive bioinformatics analysis, making this quite a complex workflow. Now, mislabeled or swapped samples can impact your laboratory by wasting those very expensive reagents and resources, causing delays of results as you try and investigate an unusual result. The integrity of your laboratory and the integrity of your results, and of course, damaging your reputation. So the MassARRAY system has a variety of sample integrity panels that allow you to quickly check both the quality and also the authenticity of your results at a low cost per sample, ensuring that you are not damaged in your reputation due to these causes. So two primary sample integrity products that I'm going to talk to you about today are our Exome QC panel and Sample ID panel. So starting with Sample ID interrogates both intronic and exonic regions with 44 targeted SNPs. We have 5 DNA quality control markers in the assay, determining the number of amplifiable DNA copies, and we also have 3 sex identification markers. And this panel has been coupled with whole genome sequencing and targeted sequencing, where those targeted SNPs are included in the targeted sequencing. Now, the Exome QC panel differs slightly in that it targets only exonic regions and exonic regions that are commonly in things like exome sequencing or targeted exome sequencing. There is 21 targeted SNPs in this panel. And because this panel is targeted to highly degraded FFPE tissue samples, we've included an extended set of quantification controls or DNA quality controls with 25 controls in this panel. Also having 3 sex identification markers. And what this does is it measures the number of amplifiable DNA copies across a broader template size from 100 base pairs up to 500 base pairs. Again, as I said, paired with exome sequencing or targeted exome sequencing. So for the iPLEX Pro Sample ID panel, the SNPs that have been selected are those that have been shown to be very well suited for the purposes of human identification. In fact, the vast majority of those SNPs come from a consortium that's been in existence for some time called the SNP for ID Consortium that published a list of 52 SNPs that can be used for human identification. So all of these SNPs have high minor allele frequency across the various different populations. So 30 to 50 SNPs has a very high power of discrimination. So 44 SNPs will deliver 1 times by 10 to the minus 18 or 1 in 5 quintillion. I didn't know that was a word, but 1 in 5 quintillion. Which is a significant power of discrimination. Now, if 60% of the markers are informative, or around about 25 SNPs, that gives 1 to the power of 10 to the minus 11, or 1 in 45 billion. And of course, the current human population is 7.9 billion. So we've got significant power of discrimination in this panel. Now, the Exome QC panel also has high minor allele frequency SNPs across all of the various different populations, and it includes 21 exonic SNPs. So in exome regions that have been selected via our selection criteria. So, 21 exonic SNPs still giving in the vicinity of 10 to the minus 9 and 10 to the minus 9 to 10 to the minus 10 power of discrimination, significantly higher than the total human population. And this panel quantifies your amplifiable template copies in a broader range from 100 copies to 100,000 amplifiable copies. So that's the equivalent, if it's all intact, of 0.3 to 300 nanograms of sample, and determines the number of amplifiable copies across a broader template size from 100 base pairs through to 500 base pairs, so that you can identify whether this sample has sufficient quality for a short amplicon technology or a longer amplicon technology based on the quality of the sample. So utilising our sample integrity panels in an NGS workflow, I'm going to show you a couple of examples where our customers have done this and have various different setups depending on the structure of their laboratory and the structure of their incoming samples. But generally, the incoming sample can either be split, or you can get an orthogonal sample such as a buccal swab in addition to the clinical samples, which can be extracted separately. The sample can be split onto the next generation sequencer and also onto the MassARRAY to generate that SNP profile. And then in your bioinformatics pipeline, you take the output from the MassARRAY system, which is a very simple CSV file, in addition to our user-friendly reports. It does output as a CSV file, so that you can then compare those SNP profiles to the data from your next generation sequencer, ensuring that the NGS reads that are coming from that sample that you believe is from that patient actually matches from a genetic barcode point of view. So you can compare those results, and if they match, proceed with reporting out your results. And if they don't match, recommend you doing an investigation on that, because clearly the results are not from the same original sample. And this gives you the confidence that your laboratory is reporting the results from that original sample that was received. So protecting your laboratory. This solution allows you to add robustness, catching labeling errors and sample mix-ups in your laboratory. Accuracy, ensuring that your results match the specimen that was received. And it gives you confidence in your reliability, in that we've tested the quality of the sample up front to ensure that it has sufficient quality for a robust and reliable result going back to your referrer. So let me talk you through several different customer success scenarios. So the first customer profile that I'm going to walk through is Ambry Genetics. So Ambry provides sequencing-based panels and solutions for a variety of different applications: oncology, cardiology, neurology, general genetics. They have a variety of different workflows depending on the markers that are being analysed, whether they're sequencing for SNPs and small indels, a separate workflow for looking at indels, duplication, structural variants. So they receive specimens from healthcare providers and medical facilities across the nation, a variety of different referring physicians across a broad geography. So once they receive the sample, accession and extract, they then split the sample into their various different workflows in addition to the MassARRAY's sample ID panel. Then comparing those results to make sure that the SNP profiles match across the various different workflows, and that all of those results are coming from the same original sample. And of course, if there is a mismatch identified, they stop at that point and do an investigation. So this allows them to have a high level of confidence in what they're providing to their clients. So if your laboratory is something similar to this, we do have a webinar by Ambry Genetics, which goes into more detail of how they've implemented our sample ID as part of their NGS workflow. That webinar is available on the Agena Bioscience website, so I recommend you going across there and you can see a more detailed description of how this has been implemented. All right, in our second profile, this is a very different situation. So this is a nationwide sequencing project, and what has happened here is that a single central laboratory has been designated as the sequencing laboratory for a variety of different clinical laboratories across the nation. The aim of that sequencing laboratory is to sequence 500,000 whole genomes within the next couple of years. And they use a similar platform for both diagnostics and research work. So diagnostic laboratories send their samples in for sequencing, researchers send their samples in for sequencing, and it all gets sequenced in a single central facility. And they're doing a variety of different both diagnostic analyses, but also a variety of different population health and longitudinal studies for clinical research. Now, the concern here is the clinical laboratory is now sending out their sample into a centralized sequencing laboratory. And those clinical labs wanted to ensure that the results that they get back match the sample that they originally sent out. So each of those laboratories that's sending the sample out is using the MassARRAY ID panel. And it's actually an exome QC panel because they're doing exome sequencing here. But they're using a MassARRAY ID panel to barcode the sample before they send that sample to the central sequencing laboratory. So instead of the central sequencing laboratory looking after their chain of custody, it's the clinical referring labs that are looking to ensure that the results that they're getting back from the sequencing laboratory and reporting to their clients are accurate. So they're barcoding them with exonic SNPs that then are also included in the NGS. And when they get the NGS data back from the service provider, they're then matching those samples up and ensuring that they come from the same original sample as they sent out. All right, so Agena's iPLEX Pro sample identity products. All of our sample identity products, whether it be sample ID, exome QC, or some of the other ones that I'm going to talk to you about later, which are soon to come, They are all single well testing panels. So one single reaction, one single well. In a 96 well plate, you can run up to 96 samples. In a 384 well plate, you can run up to 384 wells, 84 samples. So being a single well provides that inexpensive low cost per sample. The panels themselves determine the sample identity via those SNP barcodes, but they also determine the amount of amplifiable DNA template, and also the quality of that template, whether it's degraded and so forth. It can easily integrate into your lab workflow with a less than 8 hour turnaround time, very short hands-on time for these panels, less than an hour hands-on time for a plate. The results come out as both a highly interactive, user-friendly HTML series of reports, but also a CSV file or comma-separated value file so that you can put that into your LIMS system for automated matching. And they have a low amount of DNA input, and I'll describe that in one of our later panels, our Liquid IQ panel, where the amount of DNA is highly limited. So you don't want to use all of your DNA for QC and ID. Of course, you want to leave the majority of your sample for your actual analysis. All right, so with our sample integrity panels, you can verify your sample identity. If you're verifying in combination with a next generation sequencing workflow, you can take that barcode, match it to the results from your NGS output, ensuring that the NGS output is from the correct original sample, protecting you against sampling handling errors and catching any potential sample mix-ups or labeling errors that happened in your laboratory, so that you can confidently say, this is the results for the sample that I received. You can authenticate your results, doing that matching and verifying those results with high confidence, and have a very clear chain of custody documentation for your laboratory. And you can minimize those wasted reagents and the time spent for staff manual review by having an automated barcoding and identity verification pipeline. I've had situations of suspected sample mix-up in my laboratory, and the amount of time it takes to track back and do the investigation is quite significant. So this panel has allowed us to minimise to a significant extent any investigations that we've had to perform, because we've got that confidence in our workflow. So with that, thank you for attending. Let's have a quick look and see if there have been any questions come through. Okay, so we have a question. Rather than NGS, for those using MassARRAY mutation panels, HS and non-HS custom, would the identity markers be incorporated as an additional well on these panels to be compared back to the testing of the extracted DNA? Right. So if for example, if you weren't running NGS and you were running a MassARRAY panel, then of course you run the sample ID panel at the same time. It allows you to determine the number of amplifiable molecules, any PCR inhibitors, the level of fragmentation, and yes, you would run that. You would run that in addition, side by side to your mutation panel. Now, some laboratories decide to run it up front. So run the QC up front and determine which samples should go on to subsequent analysis. But if you're running the MassARRAY panels, we do have very short amplicons, and we're very highly robust on even pretty highly degraded samples. Now, we're not wizards, we're not magicians. If you don't put enough molecules in, then to achieve the desired sensitivity, then there's nothing I can do. So you should still run the QC. But you can either run it at the same time as your mutation detection panels, because it is the same workflow, and then just check, because you should have a very small number of samples that actually don't meet the quality criteria. But alternatively, you could run it up front to try and save that small number of samples to actually being run and incurring the reagent costs. And the same follow-up from the same participant. And if this could be used as an additional well on MassARRAY mutation panels, could this detect low-level contamination in addition to sample swaps? So yes, it can. Now, with contamination, you will see that the allelic frequencies are skewed, and therefore. Therefore you'll know that the sample is contaminated. What it will likely look, what it looks like on your report is that you'll get a low call number of the SNPs. Although you have high copy number of DNA, you'll have low calls because the allelic frequencies of those contaminated samples fall outside of the confidence intervals of what would be a normal Mendelian inheritance pattern of, you know, homozygote, heterozygote. So that will flag you. We do have additional panels if this is a significant problem in your laboratory. We do have additional panels which are focused on detecting low-level contamination in samples, which we can talk to you about offline. But yes, these samples will detect a contaminated sample in addition to a sample swap. So that is where you've got a mixture rather than just 100% pure from a different individual. Right, what information is available in the report outputs? Okay, so in the report outputs, I showed you some examples of the HTML, which gives you the simple summary, how many SNPs were called, amplifiable copies, whether there's expected and unexpected matches and mismatches, and so on and so forth. But all of that data and the underlying raw data comes out as a CSV file. So you get the genotypes for each of the individual SNPs in a CSV file, the sex markers in that CSV file, the number of amplifiable copies in that CSV file, and then for liquid IQ, the other parameters like white blood cell contamination and so on and so forth. So very simple, one sample per line, genotypes in each column, number of amplifiable copies in each column in a standardized format so that you can put it into your LIMS system if you're using that for your analysis and your matching. And that answers the next question, actually. Are the reports compatible with LIMS? Yes, we have a standardized CSV output that therefore you can use as an input to your LIMS system. We can actually designate the folder that CSV output is read to so that your LIMS system can pull from there.